Good evening, everyone. This is your sixth week, and in this problem solving session, we'll be talking about the topics which you have covered in week six, and then we'll also practice MCQs and we'll also look into the current literature that what is going on, and then I'll take your doubts about the assignments or other topics. So, this week six was entirely about the NMR. So when we talk about NMR, it is nuclear magnetic resonance spectroscopy, and it is a powerful analytical technique which is generally used to characterize the organic and biological molecules. So it is based on detecting the carbon-hydrogen frameworks within their structures. So in X-ray crystallography, as we have seen, it was based on the ionizations. So when electron was ionized, then the diffraction pattern was achieved. But in this case, it's not based on ionization. In this, we'll simply look into the carbon and hydrogen frameworks inside the structures of proteins in this case. So it provides valuable insights into the physical and chemical properties of the atoms and molecules, due to which it is an essential tool. That's why it is used in the research. And now there are few applications of this NMR, such as it is used in science and technology. For example, in hospitals, it is used for the MRI, which plays crucial role in diagnostics. So this is the representation of MRI. Then in pharmaceuticals and medicines also, NMR is used for designing drugs and structural analysis of biomolecules. It is also used in food industry for quality control. So with the help of NMR spectra, we can also look for the adulterations, or we can check that are there some impurities being added into some materials. So for that purposes, it is used for quality control and detecting adulterants and also ensuring the authenticity of a product. Along with this, it is used for the microscopy and material science, in which it helps to study the composition and behavior of various materials. So one of the key advantage that NMR is being used is that it is non-invasive and non-destructive. So due to its non-destructive nature, we can use the sample again and again. So that is one of the advantage, and it is also non-invasive. So it does not harm the biomolecule which we are analyzing. Now let's look into more detail into the NMR spectroscopy. So it allows us to analyze the molecules at the level of atoms. So it is used for the atomic level analysis, which allows the study of each atom within a molecule, enabling the selective probing with high sensitivity, because as it is based at the level of atoms, so its sensitivity is quite high. For example, in this case, as we can see that we can also look for the intensity of hydrogens present in some biomolecule, as well as we can also look into the carbon. So in later slides, we'll see that which of the atoms are NMR active and which are NMR inactive. So we only study those elements which are NMR active. So carbon and hydrogen. So means we will also check that which which isotope of carbon is NMR active. So those elements we can study. So in this way, they provide atomic level resolution of a biomolecule. Then it is helpful for comprehensive molecular insights. So like other techniques which focus on molecule as a whole, such as X-ray crystallography. So in that case, we have to crystallize entire protein, and then we were observing the entire protein structure. But in this case, we do not have to focus on entire molecule. Instead, we are focusing at the level of atoms, atomic interactions we are focusing on. Then versatile experimental conditions. So as we have seen that NMR is non-invasive and non-destructive. So due to that, we can means change the experimental conditions. We can vary temperature, pH, and then study the biomolecule. So for example, in this case, tryptophan is present in one of the proteins. So we are observing this tryptophan peaks NMR spectra at different pressure ranges to study that whether the pressure has any effect on the structure of a protein. So in this example, they have shown that when the protein is in folded state and whenever it is in the unfolded state. So how the peak intensity is changing. So with increase in pressure, so towards upside, the pressure is increasing. So when the pressure is increasing, the intensity of unfolded protein is also increasing, and the folded protein intensity is decreasing. So which shows that whenever the pressure is applied, the protein is getting unfolded. So with this, we can compare at different pressures that how the protein is getting unfolded or vice versa. So in this way, we can means change the conditions and study the biomolecule and their behavior. Similarly, we can also perform the pH dependent studies, in which we can change the pH conditions, and then we will see that how the protein is behaving. So in pH dependent conditions, what can happen is that there are certain aromatic amino acids. But in this case, we have considered tryptophan. So we are observing the peak of tryptophan as well as tyrosine. So tyrosine is the aromatic amino acid. So in this case, when we are changing the pH conditions, so at the higher pH values, this peak is shifting towards the right side, which means that the ppm, which is the chemical shift value, that is decreasing. So in the later slides, we'll also look into the what is chemical shift and how it varies. But from here, we can see that with changing pH, we can also see that how the molecule is changing its behavior. So as in this case, by varying pH, the peaks or the chemical shift which a molecule is showing, it is changing with change in the pH values. Then comes non-destructive advantage. So NMR is a non-invasive method which allows the repeated analysis of sample without degradation. So due to which we can use it for long times and we can continue the analysis of the same protein molecule which we have taken. So this is one of the advantage. Now let's look into another advantages of NMR spectroscopy. So other advantages are the quantitative analysis. So with the help of NMR, we can also look into the quantities. That means we can compare two different samples and then check for the quantities, that whether there is change in the quantities among both of them. So with the quantitative analysis, we can also compare that, means if we observe certain peaks which were not previously present, so this could give us a hint that there is some kind of impurity which has been incorporated by mistake or by any reason. Something is wrong in the material which we are now studying. So in this case, the blue bars, these blue peaks, they represents the adulterant or the molecule with impurities, whereas the black one is showing the peaks of the molecule which do not have any impurity, which is the reference protein with which we are comparing both of these. So in this case, this particular peak is showing that there is impurity in this sample because other peaks are aligning well with our reference sequence or the reference molecule which we have studied earlier. So with this, NMR can be used for the quantitative analysis, which can help us to detect the impurities and also check the temperature dependent behaviors. So we can compare different molecular insights. Next thing is to study the molecular dynamic behavior. So in X-ray crystallography, when we were studying, so we were crystallizing a protein, due to which the protein do not have that much flexibility. It could not freely move as it move inside the body. So in case of NMR, as we are performing the analysis in the sample form, so in this case, we can study the dynamic states of a protein. It can adopt multiple conformations. So this is also one of the advantage. So in this case, as we can see that means with the help of NMR, not only the structured, properly folded protein we can study, but we can also look for the regions which are intrinsically disordered or which do not fold properly into some structures. So we can also look for those regions as well. And we can compare among different conformations of a same protein with the help of NMR. Then comes time scale sensitivity. So with the help of NMR, we can also capture that in different time scales, how the molecule is changing its behavior. Why either it is acquiring flexibility or either some new interactions are being made or how the reaction kinetics is proceeding. So in this case, uh, means there are certain interactions which happen in some time period. So for example, this Fs means per milliseconds. So which means 10 raised to power minus 15 seconds. So this is very smallest unit of time. So in means as we move towards the left side, we are going to means smaller time units. So the interactions such as bond vibration, methyl rotation, side chain rotamers, or loop motions. So they happen in a very quick, they are very quick motions. So we need to capture them in a very short interval of time. Whereas there are global motions, which are happening in the time range of seconds and milliseconds. So they are larger domain motions. So in this case, we can also perform the time scale analysis by using NMR. So with which we will be able to identify the local flexibility or local motions as well, because these motions are happening in very smaller interval of time. So that is one of the advantage of NMR. Now, before moving further, we need to understand that what is spectroscopy, because NMR spectroscopy means that something has to be done with the light. So now we have to understand that what is the principle of spectroscopy and what is all happening in NMR. So before moving on, we need to understand that how the light is interacting with matter and how the image is formed or how we detect some object. So the study of how different forms of electromagnetic radiations interact with atoms and molecules, that is known as spectroscopy. So in NMR, we will see that how in NMR we use the radio waves, which comes under the electromagnetic spectra. So in that case, the electromagnetic waves, those radio waves, they're interacting with the protein molecules. So with that, they are means performing some kind of, they are giving us the results in form of peaks or intensities. So that is known as spectroscopy, where we have to use the electromagnetic radiations. So there are different types of light interactions, such as transmissions. So transmission is a phenomena in which light is directly passing through a material. It's not even lost, it's directly passing through it. Then the second interaction is reflection, which means that there is some surface. Whenever light is hitting that surface, it is getting bounced back. So it's not entering further, it's getting bounced back. Third interaction is refraction, which means that whenever the light is striking on the surface, it is bending by some angle and then it is going inside so that we can capture it. Then comes diffraction. So in X-ray crystallography, that was based on the concept of diffraction. So in diffraction, the light is spread out after passing through an opening or the edge. So there is a small opening from where the light is passing, and then it is scattering into different directions. But this scattering is an ordered scattering. Then comes absorption, in which the light is hitting a surface and it is getting absorbed by the material. Then the last interaction is scattering, in which the light is redirected in different directions. So light is hitting this surface and it is getting redirected into multiple directions and it is unordered. So that is the difference between diffraction and scattering. Now the best example of spectroscopy is the human eye, in which whenever the light is passing through the eye, we are able to visualize different materials. So human eye function as a natural spectroscopic system which detects the light and process the visual information. So now we'll see that how it is happening. So in our eye, we have a lens which is helpful in passing the light through it and refracting that light. So first of all, light is passing through the clear lens which is present in our eye, and the iris is the colored part, which is whenever it responds to the light. So whenever there is bright light, then it get narrows down, and whenever there is darkness, then it widens. So depending on that how much light is coming, it is trying to protect our lens. So that is one of the advantage of iris. And then cornea is the entry point. So this is the cornea and this is the entry point from where the light will come, and iris is managing that how much light is to be passed through the lens. Now the light will pass from the lens and then it will hit the retina. So retina is a membrane kind of structure which is present at the end of the eye. It's like a screen on which that particular light will means light will hit, and then that light will be converted into an electrical or the chemical signal which is further processed by our brain. So these optic nerves, they will collect that signal and it pass on it to the brain, and now the brain will convert those signals into an image. So this is how the eye is used, and it is based on the spectroscopic principles. Okay, so now we'll also look into the electromagnetic radiations. That what do we mean by electromagnetic radiations? So why we say electromagnetic? Because it is made up of two components. One is oscillating electric field and another one is oscillating magnetic field. So it has two components, and both of these fields, they are perpendicular to each other. So in this case, the electric field is represented by these red waves and the magnetic field is represented by these purple waves. So they are perpendicular to each other, and this is the direction of propagation. So propagation means to which direction it is passing. So now we'll look into other terms which are associated with these radiations. So the first term is wavelength. So wavelength is the distance between two nearest parts of the wave in the same phase. So for example, if we compare this particular point from where this wave is starting, then towards this particular point, which is the two nearest parts where this wave is in phase. Or we can also compare the crest of the means first crest with the immediate successive crest which is coming in the next region. So in this case, these 2 points are also in phase and these are successive points. So this is also referred to as wavelength. So simply it is the nearest parts which are in phase. So that is known as wavelength. And then comes frequency. So frequency is defined as number of times the electric field oscillates in 1 second. So how many times it is oscillating in 1 second? For example, in this case, this entire area represents the time interval of 1 second. So our first wave, which is red one, it is oscillating 3 times. So in this case, its frequency is 3 hertz, which means there are 3 oscillations in 1 second. Similarly, the blue wave is oscillating 6 times. So in this case, it has a frequency of 6 hertz. Similarly, the other wave, this green wave, it is oscillating 12 times. So which means it has the frequency of 12 hertz. Okay, so the unit of frequency is hertz, and 1 hertz is equals to 1 cycle per second. So similarly, 2 hertz will mean 2 cycles per second. So similarly, it will tell you the number of oscillations happening per second. And now we'll look into the relationship between the wavelength and frequency. So the relationship is given by V is equals to C by lambda. So this V is mu, which means frequency. So it does not appear as mu, but this is mu. Mu is equals to C by lambda, where lambda is wavelength and C is the speed of light. So this is the relationship between the wavelength and frequency. And now the concept is of the photon. So electromagnetic radiations, they consist of a discrete packet of energy, which are called as photons. So this is a single photon. So in this way, in this case, there are 4 photons, and each photon is a single packet which have both the electric and magnetic field. And the energy of photon is given by h nu, where nu is the frequency. And then if we put this relationship between the frequency and wavelength into the equation, then it becomes hc by lambda, where h is the Planck's constant, c is the speed of light, and lambda is the wavelength. So this is the energy which is present in each single photon. And this is to show you the electromagnetic spectrum, so which we have seen many times. So in this case, there are different waves or different electromagnetic radiations which are present. And towards the left, the frequency and energy both are increasing, and towards right, the energy is decreasing and wavelength is also decreasing. So in our case, we will look into the radio waves which are used for NMR spectroscopy. So these waves are have longer wavelengths and low energy, whereas the X-rays, gamma rays, they are high energy, and that's why they were able to ionize the electrons inside the atom, whereas these NMR and the radio waves which are used in NMR, they do not have that ionization ability because they are low energy radiations. Yeah, so this is also the same thing. The radio waves have the lowest energy as well as the longest wavelength. And if we have to see the wavelength in meters, so in that case, its wavelength is about 10 raised to power 8 meters, which is nearly about the football field. So that is the distance of a football field. So that is the wavelength range of this radio waves. So with this, you can see that these radiations are non-ionizing radiations, whereas the other radiations which we have studied earlier, X-rays, they are the ionizing radiations, which were having the ability to ionize. So if we look at the basic constituent of a molecule, then we know that it is made up of atoms. So each molecule is made up of series of atoms, and within each atom, we have a nucleus as well as the electrons, which is present in the orbitals and which are surrounding this central nucleus. And this nucleus is composed of protons as well as neutrons. So if we combine proton and neutron, so the term which is used is the nucleon, which means the combination of protons and neutrons. And these protons and neutrons, they have two key properties. That is, they have charge. So protons are positively charged, whereas the neutrons, they are neutral, as the name suggests. And then they have spin. So there is a fundamental quantum mechanical property, which means inside the nucleus, there is some spin which is associated with these protons and neutrons. So spin is the abstract quantity, which is crucial in NMR because the NMR studies are mainly based on the spin. So it is based on the nuclear spin. So spin value of a protons and neutrons is same, which is half into h by 2 pi, where h is the Planck's constant, and the nucleus has an overall positive charge and it is associated with a spin. So in this case, there are two spin states which are possible, plus half and minus half. So in the next slide, we'll see what does alpha and beta state means. And this is the direction of the magnetic field. So under the impact of external magnetic field, the nucleus, the protons which are having some spin, it will behave in certain way. There will be generation of magnetic movement. So that also we'll see. So from this slide, you have to understand that the nucleon, which is proton and neutron, they have some spin and they have some charge. And this NMR is completely based on the charge and spin of those protons. So this is what we will explore in NMR. Whereas the electrons, they are also negatively charged and they also have some spin, but they rotate on their own axis, whereas this means, so means both are having spin. Now let's talk about the NMR active and NMR inactive nuclei, because nucleus is the main thing which is the key player in uh, NMR spectroscopy. So first thing is parallel pairing, which means the addition of spin values. So if there is a parallel spin, then the spin values will add on, and which will increase the resultant spin. Whereas if there is anti-parallel pairing of the anti-parallel pairing, then in that case it will cancel out the spin. So this is the pairing of the neutrons and the protons. So in this case, the means the objects which have the parallel pairing, they will be NMR active, and if there will be means anti-parallel pairing, then those atoms will be NMR inactive. So NMR spectroscopy relies on the nucleus which have the net spin value which is not zero. So there should be some net spin value, then only the object or that particular atom will be NMR active. If all the protons and neutrons they pair with each other, then the total spin value of the nucleus will become zero. So we also have to look into number of protons and neutrons which are present in that particular atom to know that whether it is NMR active or it is NMR inactive. So in such cases where number of protons and neutrons they pair with each other, they are same. So in that case, NMR cannot be performed on that particular atom or element, due to which that particular element will become NMR inactive. Now, how to determine that whether a nucleus inside the atom is NMR active? So each atom or each element has a mass number as well as atomic number. So if both the mass and atomic number are even, which means the nucleus is NMR inactive. So this is how we represent the element. So the topmost letter represents the mass number, whereas the bottom letter represents the atomic number, and this X represents that particular element or atomic symbol. So in scenarios where both the atomic number and mass number are even, so in that case, those elements will be NMR inactive. And if atomic number or the mass number is any one of them is odd or both of them are odd, then in such scenarios, that element will be NMR active. And the commonly studied nuclei in NMR are hydrogen, carbon, so C13 isotope of carbon, then P31, N15, fluorine 19, and 29 silicon. So these are some of the elements which are used to study NMR, and because they are NMR active, because when we look for their atomic numbers as well as the mass number, so then they are either one of them is odd or both of them is odd. So in this case, the hydrogen, which is H1, which has both the atomic number as well as atomic mass as odd numbers, so due to which it is NMR active, and it has spin value of half. Similarly, carbon 13 is also having atomic mass of odd and atomic number is even. So in this case, it is also NMR active. Similarly, deuterium is also NMR active. Whereas if we talk about carbon 12, then in this case, it has atomic number of 6 and it also has atomic mass of 12. That means both of these quantities are even, due to which it is not NMR active. So this is the way how we classify the elements into NMR active and NMR inactive types. Then comes the important concept of abundance of isotopes. So now we know that that particular element is NMR active in one particular isotope form, but the concept is of abundance, that it should also be available in that particular amount in the nature. Then only we'll be able to observe the intensity, sufficient intensity of that particular element. If it is not abundant, then it is hard to study. So there is no purpose if even if we get an element which is NMR active, but its abundance is low. So that's why we need to look into the abundance of isotopes, which means what is the natural percentage of that particular isotope. So for example, carbon 12, which we have seen, which is NMR inactive, that is 99% abundant, which means if you observe the 100 elements of carbon, then 99% of them will be carbon 12. And there is only 1% chance that you will find the carbon 13 isotope. So due to which it has low abundance, although it is NMR active, but it will contribute to NMR signals, but those signals will be very weak due to its low abundance. Okay, so now this is the list of all the nucleus, all the elements which have some spin value and what is their abundance. So you can go through this table to study that when to use which element. So in this case, for example, H1 nucleus is 99.99% abundant. That means it's highly abundant. So due to which it will give a strongest signal when we use this element, which is found almost in all biological molecules. And this is also the key molecule which is used in the MRI and MRS. Then comes helium-3. So this is used in the gaseous contrast agent for pulmonary MRIs. And then the carbon-13. So it gives well-resolved peaks, but because it has low abundance, so there will be weak signals. But it is generally used for the metabolism studies. And then this fluorine, it gives strong signals because it is 100% abundant. There is no other form of this particular fluorine. So it does not naturally occur in tissues, but is used to label drugs. Because whenever we are studying something, some biomolecule or some object, then that particular isotope should also be present in that particular molecule. Then only we'll able to study it. So in this case, the F19, which do not have any other isotope, which is 100% abundant, it is not naturally present inside the tissues. So it is generally used to label the drugs and then study the behavior. Then comes sodium 23. So it gives strong signals, but short T2 due to quadrupolar relaxations, and it is used in MRI but not in MRS. So these are some of the properties which you can go through. So now we'll move into the concept of magnetism, because in NMR, as we were focusing on the spin, so magnetism also has a role to play. It will alter the spin of a particular nucleus which is present. So nucleus is a charged particle which is in motion because it has spin, generating a magnetic field. So whenever a charged particle is moving, then it generates a magnetic field. H1 and C13, they have nuclear spin of half, behaving like small bar magnets. So as we can see in bar magnet, we have north pole and south pole. Similarly, these small nuclear spins, they also arrange them in such a way that they also generate a dipole, magnetic dipole. So magnetic dipole moment, it is a combination of spin and charge, gives the nucleus magnetic properties. So every nucleus has magnetic dipole moment. So this is the inherent property of a nucleus. Nucleus already have this magnetic dipole. But what happens if we externally apply the magnetic field? So the external applied magnetic field is represented by B naught. So in this case, we are applying the magnetic field which is towards the upside direction. So in this case, if no external field is applied, then our protons which are present inside the nucleus, they are randomly oriented. But as soon as the magnetic field is applied, they become highly oriented. So which means nuclear spins are randomly oriented previously, and then after applying the magnetic field, they either align in parallel directions or anti-parallel directions to that particular applied field. So as external magnetic field is applied, so there will be some generation of energy levels between those two states. So parallel alignment will generate a lower energy state, which is more likely, in which the spin of that particular nuclear spin will also be towards the direction of the external field. So in this case, there is parallel alignment. That means both the direction of V naught as well as the nuclear spin is towards the same direction. Whereas in case of anti-parallel alignment, so this particular direction, so there is reversal in the directionality. So in this case, this is known as higher energy state, which is less likely because the spin aligns opposite to the direction of external field. So in the presence of strong magnetic field, the two spin states differ in energy, and as we will increase the magnetic field, the difference between those two states will also increase, so which is shown in this scenario. So in NMR, electromagnetic radiations is used to align the nuclear spin, because in normal conditions there is a spin but it is not oriented in any particular direction. So with the help of external magnetic field, we try to align those nuclear spins. We try to move it from the low energy state to high energy spin state, which is spin opposite state, because its spin is in the opposite direction to that of the external applied field. So that's why it is known as high energy state, because it is not visible. So we need to supply high energy to move to that state. So the energy difference will be there among these two states. Small energy difference corresponds to the radio frequency range of the electromagnetic spectrum, and delta E depends on magnetic field strength. So delta E means what is the difference of the energy between these two states, that is represented by delta E. And with increasing magnetic strength, the delta E value is also increasing, which means it is more harder to spin to change the orientation of the spin of this particular molecule now. And then comes spin flip and resonance. So to spin flip means that it was previously towards the parallel direction. Now it is flipping its spin. So now it is becoming anti-parallel. This is known as spin flip. So for this particular spin flip to happen, we need to supply some external energy, which is given by h nu. So del E will be equals to h nu, which must be absorbed to do this spin flip. And higher magnetic field strength increases del E, which requires higher frequency for this resonance to happen. So this is about the energy term and how the two states are created. First state is low energy state, another is high energy state. And these are also known as alpha state for the low energy state and beta for the high energy state. So spin states and nuclear spin number. So number of spin states a nucleus can have. That means that is equals to 2i plus 1, where I is the nuclear spin number. For example, in case of hydrogen and carbon-13 isotope, the I was half. So we have already seen in the table that what is the spin state of different elements. So in that case, we have seen that hydrogen and carbon were having spin state of half. So in such scenarios, the number of spin states, so spin was half. So number of spin states will be equals to 2 into half plus 1, which will be 2. So that means it will have 2 spin states. So in this case, as we have seen that two spin states, one is low energy state, one is high energy state. But it is also possible that we can observe more than one spin state, as in this case of deuterium, where the nuclear spin value is 1. So in such scenarios, when we look for the number of spin states, so 2 into 1 plus 1, which will be equals to 3. So in this case, we observe 3 spin states. So the concept of magnetic moment and gyromagnetic ratio come into picture. So magnetic moment means so nucleus is already having some spin value. So when we apply external magnetic field, then how the spin is changing, that is referred to as magnetic moment. That what is the change which is coming after applying the magnetic field. So magnetic moment is equals to gamma p. So gamma is the gyromagnetic ratio and p is the angular momentum. Angular momentum is net nuclear spin number, which is simply I. So gamma will be equals to 2 pi by h mu I, which is equals to mu h by I. So these are means formulas which have been given. So in this case, this is the orientation of the nuclear spin. So in this case, the nuclear spin is towards the top direction, and now in the presence of magnetic field, it is having some movement due to which it is moving in the angular motions. So this is the angular momentum which is taking place in the presence of external magnetic field. And when we say the gyromagnetic ratio, so gyromagnetic ratio is simply the ratio of magnetic moment to the angular momentum. So it is simply the ratio of them. So in the case where the spin is towards the direction of magnetic moment in which both are parallel, so in such scenarios, the value of gyromagnetic ratio, it will be more than 0. But in the cases where this direction is opposite, that means spin, which is natural spin of a nucleus, and the magnetic moment, if they both are anti-parallel, so in such scenarios, the value of gyromagnetic ratio will be less than 0. So it is simply the ratio of magnetic moment to the angular momentum, and angular momentum is the nuclear spin number. So which is the normal nuclear spin, and magnetic moment means in the presence of external magnetic field, how the spin is changing. So that is what the gyromagnetic ratio is telling you. So effect of magnetic field on magnetic dipole. So magnetic dipole interacts with the external magnetic field, and interaction energy is given by minus mu into B naught. So minus mu dot B naught, which is the dot product of both of them. So magnetic moment, which we have seen here, which is gamma P, and the field B naught, which is the external magnetic field, they can be decomposed into three components towards X, Y, and Z axis. So in this case, the energy will be mu x bx plus mu y by plus mu z bz. So this is coming from this particular equation. We need to add them because we are decomposing it into three components. So if the magnetic field is applied only along the z-axis, so in such scenarios, x and y components will become equals to 0. So the net value will be E is equals to minus B naught gamma I z. So this is nothing but mu value. We are changing the value of mu. So B is constant, and instead of writing mu, we are writing gamma P. So P is the angular momentum, which was equals to net nuclear spin, which was I. So in this case, IZ means towards Z direction, what is the nuclear spin, and gamma is the gyromagnetic ratio. So this is how we are finding this particular magnetic field along the Z axis. So the energy along the Z axis when magnetic field is applied. So spin states and energy levels. So as we have seen, there are two energy levels. when the external magnetic field is applied. So spin state corresponds to discrete energy levels. So in case of H1 and C13 nuclei, it have two spin states because we have seen by the formula of 2I plus 1. So it has two spin states, one is plus half and another one is minus half. So this is how two spin states are present. This means the possible values of Iz will be half h by 2 pi or negative half h by 2 pi. So these are the two possible I values, and where h is the Planck's constant. So in this case, the energy separation between two spin states depends upon the magnetic field. So as the magnetic field increase, this difference will also increase. And why is it so? Because delta is directly proportional to k b naught. So where it is directly proportional to b naught. So when the b naught will increase, the del E will also increase. So this is the relation between both of them. And then del E is also equals to h nu. So because energy is equals to h nu, so del E will also be equals to h nu. So with which h, where h is the Planck's constant. Now let's talk about the energy of interaction. That what will be the energy when this interaction will happen between the normal nuclear spin and the external magnetic field. So substituting Iz into the interaction energy formula. So Iz is half h by 2 pi. So if we include positive spin, positive state, then it will become equals to E is equals to minus B naught gamma half h by 2 pi. And if we add the negative spin value, that is this negative state, which is high energy state, then the energy will become equals to B naught gamma and half h by 2 pi. So because in the formula we have negative V naught, so that's why in the positive state the energy of interaction is negative, whereas in negative in spin state the energy is positive. So this is just substituting the value into the equation. This particular energy state where the value is negative, it is the lower energy state or the ground state where the spin was positive half, and it is also known as alpha state, which is most stable state. Then comes high energy state, which is also known as excited state, where spin was negative half, and this is known as beta state. So in this case, the energy value is positive. So energy difference between two states is del E, which is equals to B naught gamma h by 2 pi. So this energy difference is simply calculated by performing the difference among both of these energies. So if we subtract both of them, then we'll get this value. Then comes resonance conditions. So for absorption to occur, the applied external energy, which is h nu, must match the energy gap. Then only the spin will happen. So for particular nucleus to if we want to change the spin of particular proton or neutron, then we need to supply that particular energy with which it can spin to this particular state. It can reach this particular state, so which is known as resonance. So in such scenarios, the h nu, which is external energy, that should match the energy gap. Then only the spin will happen. Yeah, then only transition will happen to the higher energy state, and the formula for that external energy, it should be V naught gamma h by 2 pi. So then comes the concept of strength of magnetic field. So most common way to indicate the strength of magnetic field is to give in terms of Larmor precision frequency. So with the help of Larmor precision frequency, we can indicate that what is the strength of external magnetic field. So Larmor precision frequency is a frequency at which the nucleus precesses around the magnetic field, and this is given by omega 0, which is equals to nu because it is a frequency, and which is equals to gamma b naught by 2 pi. So we already seen that mu is equals to, so in the previous case we were looking that h nu is equals to b naught gamma h by 2 pi. So in this case, if we cancel h from both the directions, then nu will be equals to b naught gamma by 2 pi. So this is what we observe in the Larmor precision frequency. So because it is the frequency, but it is represented by omega naught, and this is the formula how it is derived. And this frequency will tell you that how the external magnetic field, what is the strength, how it is bringing about the change in the frequency of this particular spin movement. So this is about the Larmor precision frequency. And here V naught is measured in units of Tesla. So external magnetic field is measured in the units of Tesla or Gauss. So 1 Tesla is equals to 10 raised to power 4 Gauss. If we put the values of gamma and V naught, which is typically used, we get the mu, which is the this angular momentum, and which is in the units are megahertz regime. So the point here is that resonance frequency, which is mu, depends on the magnetic field directly, because mu is equals to gamma b naught, which means there is a proportionality, there is a relationship between them, and they are directly proportional to each other. So magnetic field which we use in NMR is really order of magnitude higher compared to what's the Earth's magnetic field. That's why this particular magnetic field is able to bring about the change. Otherwise, if it is similar to that of the Earth's magnetic field, then it will not be able to impact the spin of the nucleus. So this is what we can understand from this slide, and this represents that how the B is the external magnetic field. So at B1 value, the energy barrier is less, which is the difference between both the state is less. And as we increase the B naught, the energy difference between both the states increases, which means that higher the B naught, higher the energy which we need to supply to bring about the change in the spin states. And this omega naught represents the Larmor precision frequency, so which shows that at what frequency the spin is happening. So this is the neutron or nucleus which has the spin axis. It is rotating on it's spinning. So in external magnetic field, it will follow this particular circular motion, which will be in the form of a conical path which it will take. So this is known as Larmor precision frequency, and it will tell you that what is the strength of magnetic field. Because if the strength is high, then the frequency will also increase. So now we will look into the components of NMR spectrometer. So till now we have looked into the concepts of spectroscopy and what are the principles and what are the elements which we can use. So now we will proceed into the NMR spectrometer, that what are the components and how we can study it. So main components of the NMR spectrometer are superconducting magnets, which generates very strong magnetic field, which is greater than the Earth's magnetic field, because this is the main condition. Then only they will bring about the change in the magnetic moment. So that's why they have strong magnetic field, and the sample is placed inside the field and exposed to the radio waves. So this represents the superconducting magnets. So this is the superconducting magnet which is generating the magnetic field. Then comes second component is the spectrometer or RF transmitter and receiver. So which is transmitting the radio frequency waves and also receiving it in the end after it's coming back from the molecule. So these RF waves excite the nuclei in the sample and detect their response, that how the nuclei is behaving, how the spin state is being changed, and what is happening. So that is the work of spectrometer, which will detect for the changes. So this is representing the spectrometer. And then the last component is the computer, which is controlling the instrument and which is processing this data, which is coming from the spectrometer. So now we'll look into the working of this NMR spectrometer. So how it is working. So firstly, we'll place the sample in the presence of magnetic field and excited with the RF pulses. So in this case, we have shown the superconducting magnets which are present, which will generate a magnetic field. And this is the sample which is placed in between that magnetic field. And now we also providing the pulse of radio frequency waves. So firstly, the RF input is provided, and then this RF wave is also means how this wave is changing. So its output is also means detected by the detector. So detector is collecting the output of radio frequency waves, and then they are amplifying the signal, and then ultimately NMR spectrum is obtained, which will check that how the particular sample will behave, how it is behaving in the presence of external magnetic field. So there are some Fourier transformation which need to be performed to generate this spectrum, and this pulse sequence which is repeated multiple times, so that we can increase the signal to noise ratios, so which is SNR. So generally, to avoid the noise, we need to take it again and again, so that we should be sure that whatever is coming in the spectrum, that is not a noise, that is not. So to be confident, we need to perform it multiple times so that we can increase this. So now we will look one by one into the each component. So first thing was superconducting magnet. So this magnet is an electromagnet which is made from coils of superconducting wire, and they must be cooled to cryogenic temperatures during operations. So to maintain the cryogenic states, they are generally filled with the liquid helium, which is represented by these green wires, as well as liquid nitrogen, which is represented by this particular red circle. So means multiple liquid gases such as liquid helium, liquid nitrogen, they are provided so that the temperature conditions of this particular supermagnet, it could be cooled down. And in its superconducting state, the wire has no electrical resistance. So that is the difference between a conductor and a superconductor. So in this case, there is no electric resistance, and because temperature is also maintained at very low level, so means temperature is very low, so due to that reason also the resistance will be low, and therefore it can conduct much large electric currents than the ordinary wires and creating greatest magnetic fields. They are also cheap to operate because no energy is dissipated as heat in the windings, because if there is some resistance, then only heat will generate. But as in case of superconducting magnets, first of all, we are maintaining in the cryogenic state, and then there is no resistance. So there will be no heat buildup, due to which there will be no energy dissipation. So that is why it becomes cheap. In NMR, the superconducting coils, they are made up of niobium titanium or niobium tin alloys. So this is the constituents of the coils which are used, and these wires are wound miles long with the uniform diameter throughout, and the magnet is rested on the vibration free legs to avoid the vibrations from the ground to be transferred to the magnet. And the higher the field strength, higher is the sensitivity of NMR and higher will be the resolution. So these are some of the conditions, and this also shows that why we are using the superconductors and what are the conditions which need to be satisfied. So this is showing that how we are using the superconducting magnets and how the change in the means if we use higher field strength, then sensitivity will also increase. Second thing is probe, which is also the component. So the probe is the part of NMR spectrometer that does much of the work in terms of exciting the nuclear spin and detecting the NMR signal. So the probe goes into the center of the magnetic field, because in this probe only, because that will be in the center of the magnetic field, and this is only the thing which is holding our sample. So it will go into the center of the magnetic field and sample is inserted into the probe to perform NMR experiments. So this is how we will insert the sample to this probe, and probe contains the radio frequency coils which are tuned at specific frequencies for specific nuclei in a given magnetic field. Probe also controls the sample temperature. So the probe is the component of NMR which is holding our sample, which is providing the radio frequency at particular wavelength, and which is also maintaining our sample at particular temperature conditions. So this is the work of probe. Then comes console. The purpose of console is to house the various spectrometer units in a compact arrangement, as well as providing shielding from interferences such as electromagnetic radiations from external sources, because we have seen that visible light, UV light, they also come under the electromagnetic spectra. So in this console, we are trying to protect our sample. This is such a compact unit which does not allow the another interference of other radiations coming from external sources. So this is the purpose of console. And now comes the preparation of NMR sample. So now we have seen the components of NMR. Now we'll see that how we will prepare the sample. So key points which we need to keep in mind while performing the NMR spectroscopy is that the NMR tubes which we are using, they are made up of thin glass and they must be uniformly thick to ensure smooth spinning. And chipped or shortened tube should not be reused as they can cause the imbalance. So to maintain proper balance, because we are inserting it into the probe and then there will be some spinning which is going on. So for that, there should be a proper balance. So we need to, means we do not have to use such tubes which are chipped or shortened. Then cleaning of NMR tubes. So cleaning should also be done, because if you are observing some sample and if your tube is not clean, then it will give impurities. So rinse with the solvent used for the previous sample. So because means you are using multiple samples, so in such scenarios, if you do not rinse it, then it is possible that it will bring some impurities from the previous sample. So we need to wash it multiple times, and we generally wash it with acetone. And then we have to dry these tubes upside down in a beaker or Erlenmeyer flask. So this is the Erlenmeyer flask in which we can put our NMR tube and we'll let it dry. We need to avoid heating above 50 degrees centigrade, because if there is high temperature, then it can also warp the glass. Then third thing is degassing the samples. So now till here we have cleaned our tubes, we have made sure that tubes are fine. Now we also need to make sure that the sample which we are adding, that should also be fine. So we need to degas it, which means we need to remove the gases which are present inside the sample, so that we can improve signal to noise ratios which are coming in NMR spectra. So noise here means the faulty signals which we are observing. So method to degas the sample is freeze pump thaw technique, in which we remove the dissolved gas by cyclic freezing and vacuum pumping. So firstly we'll freeze it and then we'll perform the vacuum pumping so that any external gas which is present in the sample that could be removed. Then comes ultrasonic bath. So means ultrasonic bath can also be used to remove the trapped air. So this is how we will prepare the sample for NMR spectroscopy. And now we can proceed with the analysis of the sample after degassing and cleaning the NMR tubes. Now there is another thing which is the quantity. So now everything is done, but quantity is also playing important role, that at what quantity you are using your sample. So for proton NMR of small organic molecules which have the molecular weight less than or equals to 500 daltons, so 1 to 20 mg of sample is sufficient if the proton NMR need to be performed for small molecules. And then if we want to perform the carbon-13 NMR, since carbon-13 is only 1.1% naturally abundant, so which means we need to have the larger quantities, because we have seen the proton is very abundant, which is H1 form of hydrogen, so that is quite abundant. So even if we have the smaller quantities or smaller quantities, then also it will work fine. But in case of C13 NMR, we need to make sure that the quantities are sufficiently high. Then comes concentration. So if concentrated solutions, they can be viscous and they also result in the broad signals. Whereas if the samples are dilute, then they will lead to poor signal to noise ratio and they will be masked by the solvent and impurity peaks. So this is the difference. So in this case, if your sample which you are using, if it is too concentrated, so which will also lead to the peaks which will be distorted. And if your sample is too dilute, then in such scenarios, because the solvent peaks are also getting observed, so in this case the noise level is very high. You won't be able to distinguish among what peaks are from the solvent and what peak are actually coming from the sample. So in both of these scenarios there will be poor peaks which you will be observing. So you have to make sure that the concentration should be balanced. It should not be too concentrated or too dilute. Then the another concept is shimming. So we also need to perform shimming in NMR, because so shimming means the process of correcting the inhomogeneities in the magnetic field caused by sample distortions. So in this case, when the magnetic field is passing through the sample, so there is a possibility that there might arise some distortions in the magnetic field. They are not oriented properly. So in such scenarios, the magnetic field which are acting on this particular particle or this particle, they both will be different. So by using shimming, we can make sure that this magnetic field is homogeneous. That means it is acting constantly, or it is acting means at same level on both the molecules, so which is known as shimming. And why it is important? So poor shimming results in the distorted and broad or additional peaks. So to avoid it, we perform shimming, which uses the electrical circuits to generate small magnetic field, which help adjust the main field and improve homogeneity. So for the homogenic magnetic field, we are performing shimming by using external electric circuits which are generating some small amount of magnetic field so that it can adjust the main field. So in this scenario, as we can see, both of these molecules or means particles which are present, they are experiencing the same magnetic field. And this is the representation that if means you have carefully shimmed the magnet, then in such scenarios, the peaks will be sharp and well resolved. You will be able to means find the difference between each peak. Whereas if it is poorly shimmed magnet, then the peaks will like broad and distorted. So you won't be able to distinguish. So that is the purpose of shimming. Now, how to interpret the NMR spectra? So till now we have seen that what are the components of NMR and how the sample is prepared and what are the things which we need to keep in mind while means loading the sample and all those things. Now we'll see that how we can interpret the spectra, which is coming to us in the end after performing the NMR spectroscopy. So NMR spectra represents the molecular level information. So what that spectra is giving us? It is giving us molecular level information. Different atoms in different chemical environments will make different peaks because there is difference in the chemical environment. And what chemical environment means? That for example, if we talk about the proton spectra in this case, if we talk about the H1 NMR spectra, then in this case, we will be focusing on all the hydrogens. So when we say chemical environment, which means that this particular hydrogen to which particular other elements bonds it is attached to, what are the other atoms which are attached to this particular hydrogen, that will represent the chemical environment of this particular hydrogen. So then there is a concept of integration. So the area under the peak corresponds to the relative number of protons in each environment, and peak ratio, which is A is to B is to C. So now we'll see this example. So in this case, first of all, whenever you obtain the NMR spectra, that tells you that what are the distinct or unique type of hydrogens which are present in that particular molecule which we are studying. So in this case, we observe three peaks, which means that there are three unique hydrogen types which are present. These three have distinct environments, chemical environments. So in this case, each hydrogen which is present on this particular carbon, Hc, so it has similar chemical environment. This also has the carbon which is attached to H2 and then to these particular functional group. Similarly, each of these hydrogens, they have neighbors, similar neighbors. So that's why these all hydrogens, they are means similar to each other. So this will form a one single unique hydrogen peak. Then similarly this Hb, so this particular hydrogen, these both the hydrogens, they also have exactly same neighbors. So that's why they are not different from each other. So they will also generate one single peak in this case. And then this Ha, this particular, all these three hydrogens which are present on this particular carbon, so this is also having the similar neighbors. So due to which these all three hydrogens, they will also have a single peak. Now comes sub-peaks which are present inside these peaks. So in this case, Ha is singlet with no hydrogen on the adjacent carbons. So now when we are breaking it into sub-peaks, so how we will find it? So for that, we need to find the neighbors, and then we will check that whether in the neighboring atom which is present, whether it contains the hydrogen or not. So in case of these Ha, this particular carbon is linked to carbon which is having double bond O, which is the carbonyl carbon. So it do not have any hydrogen. So that's why we will only observe one single peak, so which is known as singlet. That means there is no hydrogen present in the adjacent carbon. Whereas if we talk about the Hb, so in this scenario, the adjacent carbon is having three hydrogens. So in this scenario, it will have a triplet because there are two hydrogens, 2Hb on the adjacent carbon. In this case, Hb, when we are talking about Hb, then it has three adjacent hydrogens, and the number of peaks will be equals to number of hydrogens which are present on the adjacent carbon plus 1. So in this case, we'll observe 4 peaks. Whereas if we talk about this Hc, so in this case, the adjacent carbon is having two hydrogens. So the peaks which we will observe will be triplet. 3 peaks we will observe. So maybe in the next slide, we'll see what is the formula with which we can find out that how many peaks we will observe depending on the number of hydrogens which are present on the adjacent carbon. So in this scenario, first thing is to remember this, that how many peaks, sub-peaks will be formed and how many single peaks will be observed. Now next thing is the intensity values, that how much intensity will you observe. So this particular peak intensity depends on the number of hydrogens which are present. So for example, in this particular Hc, there are three hydrogens which are being involved. So in this case, its peak intensity will be equals to 3. Whereas in case of Ha, here also we have three hydrogens which are giving rise to this particular single peak. So this will also have the peak value of 3. Whereas this Hb, it only have two protons, so its peak value will be 2. So in this case, the peak ratio of A is to B is to C. So Ha is to Hb is to Hc is the 3 is to 2 is to 3, because Ha consists of three different hydrogens. B is the Hb, which consists of two different hydrogens. And then in Hc, we have three different hydrogens. So this is how the peak ratio is calculated for a given molecule. And this will help us to elucidate the structure, because in the peaks, when you will see the intensities, then you will be able to know that how many number of hydrogen molecules are present. So integration means the same. So integration is telling you that ratio of protons by measuring the area under each peak of the signal. So by measuring the area, it will tell you the ratio of protons which are present. That is known as integration. So in this case, the integration of A, which is CH2, is 2, because there are two hydrogens which are present, and which is obtained by means measuring the area under each peak. Similarly, for this B, which is CH3, so it has 3 hydrogens. So after integration, the peak value will be 3. So after integration, we will get the peak value of 3, and the ratio of A's to B will be 2:3. Similarly, in this case also, uh, CH3 present on these two different carbons. So these hydrogens, they are having the similar chemical neighbors, which is CHBr. So from this carbon and this carbon, all these hydrogens which are present on all, both the terminal carbons, they are having same neighbors. So in this way, both these CH3s, the hydrogen present on both these CH3s, they are same. They will only give a single peak. So this is the peak, and when we will integrate it, so because there are 6 hydrogens which are giving this single peak, so that's why after integration we'll get the peak intensity of 6. Whereas in this middle carbon, which is having only a single hydrogen, so in this case, because only one hydrogen is there, so peak value will be 1. But how many number of sub peaks we'll see? So that will depend on the adjacent hydrogens or adjacent carbon containing, sorry, adjacent carbons. So how many hydrogens the adjacent carbon contains? So in this case, the adjacent carbons contain 3 and 3, so that is 6. 6 hydrogens are present, and when we will look for the sub peak, so that will be equals to 6 plus 1, which will be 7. So in the case of B, we'll see 7 peaks. Okay, so in the next slides, you will get more clarity because we'll see that how we will analyze it. Okay, now comes the common issues in NMR spectra. So what can happen in different scenarios? So first thing is bad signal to noise ratio, which means if there will be a poor resolved peak due to noise. So in this case, because it's not properly resolved, so this is known as bad signal to noise ratio. This is the issue in NMR. So we do not, if our NMR spectra is like this, we cannot trust it because there is a lot of noise. And then the second thing is if there is improper scaling or too high concentration is present. So in such scenarios, the peaks are very tall that they will be cut off. So in this also, we will not be properly able to analyze the spectra. The third thing is signals too small. So if signals are too small, then also the spectra is not good. Or if signals are too broad, that means they're broad and not means peaks are not sharp, so that is also not a good NMR spectra. Then comes excessive peaking and distorted signal, so this is also not a good NMR spectra. And then areas without signals should be excluded. So in such scenario, if you observe proper signal in some area and some area do not have signal, so we need to exclude them and then we can proceed with that particular NMR spectra. So this shows you that what are the issues with the spectra and how the faulty NMR spectra will appear. So we need to make sure that the NMR spectra which we are using for analysis, it should be well defined. The peaks should be well separated enough from each other, so they should not be very broad or too shallow or like very small. So these are the things which we need to keep in mind while analyzing the NMR spectra. So now we will talk about the nature of NMR absorptions, because in NMR spectra we will be comparing the chemical shift. So the concept of chemical shift will come into picture. So absorption frequency varies for H1 and C13 nuclei due to surrounding electrons. So now we will see that how the chemical shift varies among different elements. So because in carbon and hydrogen, means absorption frequency will vary because there is a variable number of electrons which are present in both of these elements which are surrounding the nucleus. So local magnetic fields, electrons create a shielding fields which reduce the effect of the external magnetic field which is applied. So due to which the field which is actually acting on that nucleus will be equals to B applied minus B local. So this is how the external magnetic field is applied. So now what is happening is that there is some kind of shielding which is also working because electrons are shielding the nucleus. So external magnetic field is applied, but electron is shielding the effect. And then there will be only a little bit of effective magnetic field which will be actually working on the protons. So this effective is find out by the subtracting the B naught by the shielding magnetic field. So with this, the concept of chemical shift comes into picture. So resonance frequency relative to a standard, which is known as chemical shift, that what is the frequency with respect to standard. And this chemical shift, which is delta, which is represented by delta, so that is equals to frequency of signal minus frequency of reference divided by spectrometer frequency into 10 raised to power 6. So because in this case, both the terms in numerator and denominator are frequencies, which will be in hertz. So this will cancel out each other, hertz, and then we get only 10 raised to power 6, which is also represented as parts per million. So that's why the unit of chemical shift will be parts per million. And this is the example in which we have CH3CH2OH, which is ethanol. So in this case, we have three different hydrogens which are present. First is of this OH group, then another one is of this carbon. So both of these hydrogens, because they have same chemical environment, so both of these hydrogens will these are known as the unique hydrogen. And then this all these three hydrogens are also one single unique hydrogen. So this one is 3. And then they have find out that how many subpeaks will be present. So in this case, CH3 it is bound to a carbon which has two hydrogens. So the number of peaks, subpeaks which we will observe here will be 2 plus 1. That's why it is 3. Similarly, for this second case, the second hydrogen, this is attached to this particular carbon which has three hydrogens and the OH group where one hydrogen is present. So ultimately it has four different hydrogens which are attached in the adjacent molecules, adjacent atoms. So its subpeaks will be 4 plus 1, which will be 5. And similarly for this OH, because uh, its adjacent carbon has two hydrogens. So in this case, it will be 2 plus 1, that will be equals to 3. So there will be 3 subpeaks which we will observe here. Yeah, and now they have also shown the chemical shift values. So in this case, where the CH3 is present, so in this case, the chemical shift value is less, which is nearly about 1 ppm. And as we move towards CH2 group, because it is present close to OH groups, so chemical shift value, the OH where the hydrogen is present next to O. So in this case, the chemical shift value has increased to this 2.5 ppm. And then for this second hydrogen, which is attached to this carbon, which is one side it has OH and on the other side it has methyl group. So it has the highest ppm, which is about 3.5. So this is to show you that how the chemical shift is varying based on the type of hydrogen and to what are the adjacent chemical neighbors to which it is linked to. So we'll see that how it is working. What are the factors which are influencing the chemical shift values? So this is the basic steps which are working in the NMR spectroscopy. So firstly, as we know that whenever no external magnetic field is applied, then our means nucleus, it is randomly oriented. Protons and neutrons are randomly oriented. But as soon as we apply the magnetic field, then they will either take the parallel orientation or anti-parallel orientation, on the basis of which there will be two states which will be generated. First will be low energy state and next will be the high energy state, which will be beta spin state. And now to change the spin of the particular proton or neutrons, we need to provide some external energy, which in this case is provided by the radio frequency wave pulses. So these radio frequency wave pulses, they are bringing about the change in the spin, which is also known as spin flip from alpha state to beta state. So which is means obtained by applying the electromagnetic radiations. In this case, we are using radio frequency waves. So by which spin flip is happening, and now each type of proton will release certain energy, and which is converted to ppm, which will be the parts per million, which is the shift which is coming. So we will be in the end, the detector will detect that chemical shift. So this is how this is the principle of NMR spectroscopy, that how the external magnetic field is influencing the atoms which are present, or the neutrons or the protons present inside the nucleus, how they are changing their spin states, and ultimately that spin state is captured. So induced magnetic field will bring about shielding and de-shielding. So because in the previous case where I was telling you that this is the ppm value for different types of hydrogens. So why is it so? Why they vary from each other? So now we'll see what are the factors which are influencing it. So first thing is shielding. Shielding means the electrons which will induce a secondary magnetic field which will oppose the applied field. So till now we have seen that there is some kind of shielding which is provided by the electrons which are surrounding the nucleus. They're not allowing the complete magnetic field to pass through the neutrons or protons. So they are providing some kind of shielding which will reduce the effective magnetic field felt by the nucleus. So if there is high electron density, which means if greater number of electrons are present, so in such scenario, they will provide more shielding. So in such case, there will be upfield shift. That means the chemical shift value will be less and energy will be low. Second condition is de-shielding, which means if there is lower electron density, then what will happen? It will not be able to shield the effect of magnetic field. So in such case, there will be stronger applied field effect, so which will shift the signals to downfield, which means high del, that is high energy state. And electron withdrawing groups such as nitro de-shield electron donating groups such as alkyl. So alkyl is the electron donating group which will bring about shielding, whereas the electron withdrawing groups are nitro which will de-shield. So with this, you can understand that de-shielding will increase the value of chemical shift, whereas shielding will decrease the value of chemical shift. Now the another term is magnetic anisotropy. So what magnetic anisotropy means, that pi electrons, they generate local magnetic field in non-uniform ways, which is known as magnetic anisotropy. That means induced magnetic field reinforces the external magnetic field near the protons which are de-shielded. So in case of aromatic protons, this represents the benzene ring, and this is the circulating pi electrons which create the ring-like structure that is represented by this red line. And in the benzene ring, we also have the protons which are present. So this is the spin of proton, and this is the external magnetic field. So in this case, the direction of the spin of proton as well as the magnetic field is in the same direction. So which means that it is de-shielding it. That means it is not reducing the effect of magnetic field. That is known as de-shielding effect. That's why in case of aromatic protons, the del value is nearly 7 to 8 ppm, which is de-shielded state. But in case of aldehydes, the del value increases, which is 9 to 10 ppm, because they are strongly de-shielded. In case of alkyne protons, del value is 2 to 3 ppm, which is very less, because in this case there is shielded, because they are shielded. These alkynes are the electron donating groups, so more electrons, that means more it will shield the effect of B naught, and less will be the del, less will be the chemical shift. So which we have seen here, that in presence of shielding, the del is less, that means uh, chemical shift is less. Okay, and here is the another, to understand another concept. These are certain examples, and here are the explanations that why is it so. So more electron withdrawing the group, the stronger the protons feel the magnetic field, as they are less shielded by the electrons. So what we need? Electron withdrawing groups. So if electron withdrawing groups will be present, then we will observe a de-shielding effect and del will increase. So this is means towards the left side, it represents the downfield. Downfield means frequency is high and electrons are de-shielded. Whereas towards the left side, it is known as upfield, which means frequency is low and electrons are shielded. That means more number of electrons are present. So in this case, we are having the electron donating groups. So what are the electron donating groups? Such as this alkyl group. This is the electron donating group. So this increase the number of alkyl groups, and this will shift the signal downfield. So uh, yeah, so in this case, means this is the upfield. So we are moving towards the downfield. So what we need to do? So carbonyl carbon. So carbonyl group is electron withdrawing group, and it and the frequency of hydrogens next to the other are shifted to downfield by 1.5 and found at 2.5 ppm. So because carbonyl group is electron withdrawing, so electron withdrawing group will uh, bring about the de-shielding effect, which will move from upfield to downfield. Whereas in this case, in both of these cases, we have OR and chlorine. So oxygen and chlorine are not as electronegative. So, and the ppm value is also less downfield. So iodine, bromine, they are some of the groups which shift the signal to this region towards downfield because they have high electronegativity. Okay, so now then fluorine is most electronegative element. So it is de-shielding and shifting the signal of the adjacent protons to 4 ppm. So in this case, we have seen that how the chemical shift is increasing towards the downside as the groups which are electronegative or which are electron withdrawing groups, as they are increasing. And this is also then other examples of other groups, if they are present, then how they will bring about the change. Either they will move the molecule towards upfield, or either they will shift the chemical shift towards the downfield. Now we will look into the factors which affect the chemical shift. So which we have a little bit we have discussed it. So first thing is hybridization state of carbon. So chemical shift of hydrogen varies depending on the hybridization of carbon it is attached to. So what is the hybridization state? So the trend is that alkenes, that is RCH, they have higher del values, that means around 5 to 6 ppm, whereas carbonyl groups, which is C double bond OCH, and the alkynes, that is C triple bond CH, they appears in 2 to 3 ppm range, whereas the alkyl CH groups, which appears at lower del values, about 0 to 2 ppm. So this is the trend. So as we are moving from alkyl to alkenes, the chemical shift is increasing. This is the relationship. And then comes variation of electronegativity with hybridization. So electronegativity and the hybridization is also playing important role. So if we move from sp3 to sp, the electronegativity of carbon is also increasing. So in this way, if we talk about orbital size, then it is high in sp3, then sp2, then sp. But if we talk about electronegativity, then this order is reversed, which means if our element is in sp hybridized state, then it has highest electronegativity, and higher the electronegativity, higher will be the de-shielding. That means it will move towards the downfield, and the chemical shift value will also increase. That means there will be higher ppm values which we will observe. So this is one of the factor which is bringing about change in the chemical shift. Second is magnetic anisotropy. So in case of alkenes, alkenyl hydrogens experience de-shielding due to anisotropic magnetic fields, resulting in higher chemical shifts. So in this case, these are the alkenes where double bond is present. So in both of these scenarios, if we look at the spin of the proton, then it is towards the upward direction, whereas the magnetic field is also in the upward direction. So which means the resultant spin, they will add up and it will increase the net spin, which means the magnetic field which is acting on the proton, which will also increase. So this is known as the de-shielding effect. Whereas if we talk about alkynes, this is the alkyne. In this case, the hydrogens which are present in this position, they are having spin towards the downward direction, whereas the magnetic field is applied towards the upward direction. So in this case, they have opposite spin, so which will bring about the shielding effect. So due to this shielding, the chemical shift value will be less. It will move towards the upfield. So this is the difference between the alkenes and alkynes, and it ultimately depends on the directionality of spin. So proton spin as well as the external magnetic field spin. So based on it, it determines that how it will bring about the change in the chemical shift. So from this slide, you only have to keep one thing in mind, which means what is the hybridization state. So higher the electronegativity, higher will be the de-shielding effect, and higher will be the value of del, that is chemical shift. Second effect is inductive effect, which means what is the electron withdrawing effect. So this effect increases with great number of electronegative atoms, because electronegative atoms have higher inductive effect. So this is the comparison. In this case, we have CH4, and if we have one one I is present, so it is 2.2 ppm. If we have bromine, that is 2.7 ppm. If we have chlorine, then it is shifting to 3.1 ppm, and if we have fluorine, which is highly electronegative, so this shifting to 4.3 ppm. So which is showing that as the electronegative group means the electronegative group is present adjacent to the carbon which is having the hydrogen, then it will lead to de-shielding effect, which will increase the chemical shift value. And second thing is the effect decreases with increasing distance. So this also matters that at what position this particular electronegative group is present. If it is close to the hydrogen, then this de-shielding effect will be more. If it is lying far away, then the effect will also decrease. For example, in this case, the bromine is present next to the carbon containing hydrogen. So this is having the ppm of 3.30. Whereas in this case, that particular hydrogen which we are considering, it is having a CH2 group and then bromine is present. So now distance has increased. So for this particular hydrogen, the distance has increased. So chemical shift will not be that sharp. It will be 1.69. And in this scenario, there are two methyl groups present in between and then bromine is present. So in this case, the distance also increased from this hydrogen to Br. So due to which ppm value is quite low. So this shows you inductive effect tells you higher the electron negativity, higher the de-shielding, and higher the distance, lower the de-shielding. So distance should be as less as possible. Third factor which plays role to affect the chemical shift is the steric effect. So in this case, methyl de-shielded by the one bond. Okay, so there is one bond which is de-shielding the methyl group. In this case, methylene de-shielded by the two bonds. So there are two carbons which are present next to the CH2. So there are two carbons which are bringing about the de-shielding effect. And then methine, which is de-shielded by three bonds. So there is only one hydrogen. So in this case, increasing the alkyl substitution increase the chemical shift. And means towards the left side, we are moving to downfill. So if there are three CH groups which are present adjacent to carbon, so it will be in the upfill. As we keep on reducing the number of other C groups which are bonded to the carbon to which hydrogen we are studying, so it will reduce the effect, steric effect will be reduced. In this case, the steric effect is as high as possible. Because in this scenario, we are trying to substitute the alkyl group. We are removing the alkyl groups and adding the hydrogens or other groups, electronegative groups. Then we are moving to de-shielded place. Fourth thing is the hydrogen bonding. So in methyl salicylate, this is the methyl salicylate, which has strong internal hydrogen bonding. The NMR absorption for OH is about 14 ppm way downfill. So hydrogen bonding is also playing important role. If we have strong hydrogen bonding, then it will bring about more downshielding effect, more de-shielding effect, which will increase the chemical shift values and it will move it to the downfill. Yeah, so from this slide, the things which we need to keep in mind is about the electronegativity, distance, steric effect, less number of alkyl groups attached to the carbon. So more will be the chemical shift change, more will be the increase in the chemical shift values, and higher the hydrogen bonding, more will be the de-shielding and value of chemical shift will also rise. So these are four of the factors which are playing important role in the chemical shifts in NMR. Now the hydrogens which are present in NMR spectroscopy, they are of multiple types. So first type is homotopic. Homotopic hydrogens mean if we replace each hydrogen with different atom, they will still result in identical molecules. So in this case, D and H, so means in this place we have replaced this hydrogen with D. And in the other case, if we replace the top one hydrogen with D, so if you compare both of these compounds, both of these molecules, then in this you will see that even if we replace the hydrogen with some other atom, they are still the same. There is not much change in them. So these are known as homotopic hydrogens. So both these hydrogens are homotopic and the resulting molecules are the same since the replacement did not create a stereo center. So no stereo center is created and they will also give the same NMR peaks. So these type of hydrogens are homotopic. Second type of hydrogens are enantiotopic, which means that replacement of each hydrogen with D gives the enantiomers, which means they are mirror images. So in this case, these are the two hydrogens. And if we replace it with D, and in this case we are replacing it with D, and now the means images which we are observing in both of these cases, if they are like the mirror images of each other. So this is the enantiomer. So in the topmost, the difference among both of them is the groups which are attached to it. So in this case, it was the hydrogen which was attached in the top form. In this case, we can have any other atom which is attached to this group. So in this case, they will generate mirror image of each other. So still the stereo center is replacement creates a new stereo center, but in the enantiomeric form, the peaks which they will give in the NMR spectra, that will remain the same. Then comes diastereotypic hydrogen. So the replacement of each hydrogen with D leads to diastereomers. Stereomers means they are not the mirror images. So in this case, both of these hydrogens. So in this case, if this hydrogen is replaced with D, whereas in this case, the red hydrogen is replaced with D. So in both of these cases, they are the diastereomers because since they are alkenes with different connectivity, so there will be change in the stereo center in both of these cases. And in this scenario, because there is double bond which is involved, so this will also bring about the change in the NMR spectra which we will be observing. And the second example of this is the if we replace this hydrogen, so red and blue hydrogens we are considering. So if we replace blue hydrogen with D, and in this case, if we are replacing red hydrogen with D, so in this case also both of these hydrogens they are diastereotypic hydrogens, because if there are already stereocenters exist in the molecule and replacement creates an additional one, so resulting molecule will also be diastereomers. So this is about the three types of hydrogens. You don't have to go into the detail. This is just to understand that means what different types of hydrogens which can be present inside the spectra, so that we can distinguish, because if there will be homotopic hydrogens then we won't be able to distinguish among both of them, whereas the diastereotypic hydrogens they could be well distinguished from each other. So when does it matter? So there are two conditions. For example, in free radical fluorination, for example, if we have butane, so on the second carbon, substitution of CH with chlorine will lead to mixture of stereocenters. And it's important to recognize when this can happen. So for that reason, we must understand that what type of hydrogens which are present on that particular molecule. Second thing which is more common is the homotopic protons. They have exact same chemical shifts, whereas in enantiotopic protons, they have also same chemical shift in vast majority of situations, and they can bring about the change in the chemical shift only when they are placed in the chiral environment. So if the functional groups attached to these two positions, if they are different, then they can bring about the change in the chemical shift. Otherwise, if it is same, if it is achiral, that means both of these will become exact identical. So in such scenarios, there will be no chemical shift or no change in the chemical shift which will be brought about by those two hydrogens. Then comes diastereotypic protons. So they have different chemical shifts in all the situations, because they are bringing the new stereocenter. So they are changing the stereocenter. So that's why they will have different chemical shift values in all the conditions. So this is just the reason that why we need to understand that what type of hydrogen is present, because then only they will bring the change in the chemical shift. Otherwise, they will not bring any change in the chemical shift and we won't be able to differentiate them. Now, these are some of the means examples. So coupling in H1 NMR. So what coupling means? So coupling means the interaction between the spins of neighboring nuclei in a molecule, which may cause the splitting of NMR spectrum. So because in the previous case when we were studying about the sub peaks, so sub peaks are nothing but they are the splitting of peaks. So within each peak, there is some splitting which is happening. That is means how that splitting is happening, that is known as coupling, because there are nuclear spins which are interacting with each other, so which is bringing about this uh, splitting in the NMR signal. So what are the rules for spin-spin coupling? Only the chemically known equivalent protons show spin-spin coupling. So in this case, both this CH3 and CH groups, so the hydrogen which is present in CH3 and CH, only they can show spin-spin coupling. Whereas the hydrogens which are present in this methyl group, all those hydrogens, they will not show spin-spin coupling to one another. They all are same. So that is one of the rule. Second is protons on adjacent carbons normally couple. Protons separated by 4 or more bonds will not couple. So this is the second condition. So coupling will only happen on the adjacent carbons and not on the means carbon which is separated by 4 or more bonds. So this is also one of the condition, that whenever we are studying the coupling, then see this CH3, its coupling will only be influenced by this CH. If there is some other CH which is lying next to CH, then that will not influence the coupling. So that is the second rule, which means that only the adjacent carbon will normally couple, whereas the protons separated by 4 or more bonds, they will not couple. Third rule is the splitting pattern is related to number of equivalent hydrogen atoms at the nearby nuclei. So the proximity of an equivalent hydrogen on neighboring carbon atom causes the signal to be split into n plus 1 lines, which means that in this scenario, these three hydrogens which are attached to the methyl group, they are nearly the same. So they are attached to one carbon which has H, one single hydrogen. So this is the neighbor of this CH3. So 1H, so that is equals to N. That means N is the number of protons present on the neighboring carbon. So that is 1. And the number of splitting which will happen is N plus 1. So that means this CH3 will give rise to 2 peaks. So there will be splitting of signal into two peaks. This is the n+1 rule. Yeah, this is known as multiplicity or splitting of the coupling pattern of each signal. Coupling arises because the magnetic field of vicinal or the adjacent protons influence the field that the proton experiences. So due to which we see this splitting which is happening. Okay, so this is the example of CH3, CH, Cl2. So till now we have seen other examples as well. So in this case, there are two different types of hydrogen. One is the CH3 hydrogens and then another is the CH hydrogens. And we have also discussed that why there are only two single peaks which we will see, because all the hydrogens which are linked to this terminal methyl group, they are having the exactly identical environment, identical chemical environment. They have same distance to other functional groups which are present. So that's why these three hydrogens, they will behave equivalent, means equally they will behave. So that's why this will, all these three will form a single peak. Similarly, in case of CH, there is only a single hydrogen. So in this case also, this particular hydrogen will behave differently from these three hydrogens, because in one side it is linked to chlorine and on the other side it is linked to methyl. So that's why there will be two unique peaks which we will see. Then second thing comes about the splitting. So splitting depends upon the hydrogens which are attached to the neighboring carbon. So in case of green CH, in the green hydrogen, this is linked to methyl group on one side and chlorine to other side. So chlorine do not have hydrogen, so we will not consider it. Now we will look at the methyl group. So it has three hydrogens. So now CH, there will be spin-spin coupling between the proton of CH and CH3. So there are three protons which are present on the neighboring carbon. So splitting will be equals to n plus 1. That means number of neighboring protons plus 1. So in this case, the green hydrogen has splitting of n plus 1, that is 4. So there will be means four different peaks which you will see for this particular signal. Similarly, for the CH3 group, because there is only one adjacent hydrogen, so that will be n plus 1, that is two peaks. So this is how this is what is happening, what is coupling and why the splitting is happening. So I hope now it is clear that why we were seeing different, means although we have only two signals, but we are splitting it into individual components into between that. And this is the another example of HA and HB. So this is also the same. So I won't again discuss it. Then this is the summary of the signal splitting pattern in H1 NMR. So in this all of these examples, we were only focusing on the H1 NMR, in which we were focusing on the hydrogen. In the later slides, we'll also see that we can also see the C13 NMR spectra as well. So in H1 NMR, we will be focusing on the hydrogen. So the pattern is that N protons split the signal into N plus 1 peaks, which is known as N plus 1 rule. So in this case, if we have the example of this molecule, HA and HB. So HA is attached to carbon which has only one single proton. So this signal will be split into 1 plus 1, which will be 2, which is also known as doublet. So if there are two peaks, it is called as doublet. Similarly for HB also, it has only one adjacent proton, so it will split the signal into n plus 1, which will be 2. So there will be two peaks which we will observe. So that is known as doublet. In this example, HA is linked to a carbon which has two protons, so it will split into 2 plus 1, which means three peaks we will observe, and it is known as triplet. Whereas if we talk about HB, then it is attached to carbon having only single proton. So there is only single proton which can couple with this HB. So the n plus 1 will be 2, so there will be two peaks, that is doublet. Similarly, let's consider this particular example. So in this case, HA is linked to a carbon having having three protons. So all these protons have ability to couple their spin with this HA. So the peaks which we will observe will be 3 plus 1, that is 4. And if there are four peaks, then that is known as quartet. So these are some of the terms which you must be aware of. Okay, now we have talked about coupling and splitting. Now there is another term is coupling constant. Because if coupling is happening, then there is some constant value also which is associated with it, which is known as coupling constant. Coupling constant represents the distance between the peaks in the multiplet. So multiplet means if you are observing more than one peak, that is known as multiplet. So it is a measure of splitting effect. It tells you that what is this, how much intensity of the splitting effect is taking place. So it is known as coupling constant. It is denoted by symbol J and its units are Hertz. So in this case, if we consider CH2, so if we consider this hydrogen, it will split the signal into four different peaks because it has adjacent three protons plus one, so there are four peaks, and the distance between each of these sub peaks is known as coupling constant. So it is telling you the distance between each of these peaks. This will tell you that what is the intensity of splitting which is happening, and it also represents the effectiveness of spin-spin coupling and useful for analyzing the complex proton NMR spectra. So this is it about the coupling constant. And then if we talk about the vicinal coupling, so it refers to spin-spin interactions between two hydrogens, HA or HB, with that are three bonds apart. So till now, whatever we have discussed, we have seen the rule that if the distance is more than three bonds, then we will not, that particular proton will not couple the spin. So in this case, there is only three bond distance. That means this hydrogen and this hydrogen is separated by three different bonds. So which is known as vicinal coupling. That means adjacent coupling. And J is the coupling constant. So according to this system, coupling constant JAB represents the interaction of HA with HB. And if we talk about JBA, that means we are looking for the interaction between HB with HA. And since these interactions are reciprocal, that means mutual influence between the two protons, so the magnitude of this coupling constant should also be in the same direction. So that means JAB should also be equals to JBA. So this principle holds true if there is symmetry in the spin-spin coupling, and which is normally observed in the NMR spectra, that there is coupling in the, there is symmetry in this coupling. So that's why JAB will also be equals to JBA. So this is not important. This is just to show you that there is symmetry in the spin-spin coupling. And then typical coupling constants. So till now we have only looked into the two-bond coupling which occurs between non-equivalent hydrogen atoms on the CH2 group of an alkene, which is this one. Now long-range coupling. So sorry, this one is the alkene. And now long-range coupling means if there are more than three bonds apart. So for example, if this hydrogen which is linked to this carbon and this particular hydrogen which is present on the para position, so now they are separated by more than three bonds. So in this case, there are five bonds which are present. So among them, there is also possibility that coupling can happen. That is known as long range coupling. But the J value decreases as the number of bonds increases. So means if the distance will increase, then the J value will slightly decrease. So coupling constant will decrease with increase in the distance among the hydrogens for which we are studying the coupling effect. And another term is the Pascal's triangle in NMR splitting patterns. So the relative intensities of peaks in a multiplet are determined by the Pascal's triangle. Because if three peaks are observed, then we'll, if they are splitting into three peaks, then each of them will look for the intensities. So it is determined by the Pascal's triangle. So for example, if we have three peaks, then the ratio of their intensities will be equals to 1 is to 2 is to 1. So this is the triangle which tells you that depending on the number of peaks, this will be the ratio of their intensities which you will observe. So for example, if there will be four peaks, then there will be the ratio of 1 is to 3 is to 3 is to 1. If there is five peaks, then the ratio will be of 1 is to 4 is to 6 is to 4 is to 1. And the way how this Pascal triangle is built, it is depending on the summing up the adjacent values. So for example, from here 1 is coming. So this particular box will also have value of 1 because there is nothing adjacent to this one. Similarly, this will also have value of 1. But from now the next row, so this will have 1, and now the middle uh, place will have the 1 plus 1. So that is 2. Similarly, in this case, we will add those two adjacent boxes. So this will be 3. Similarly, here also it is 3. This is 1. So this is how the Pascal triangle is built. And this generally tells you the relative intensities. Means how after splitting, what will be the intensity of peaks which you will observe. And these are the examples. So yeah, we have already discussed. So in this case, there are two unique hydrogens, one from CH3 and one from CH2. And then the splitting will happen on the basis of n plus 1 rule. In this case also, we have three different types of hydrogens. First hydrogen is present on CH3 group, second hydrogen is present on CH2 group, and then third on CH2 group. And how they are different? Because we are comparing their environments, chemical environments. We are comparing that to which groups they are attached to. So if groups on both the sides are different, then that particular hydrogen will be a unique hydrogen. So that's why in this case we will see three main peaks, and then those peaks will further split depending on the spin, on the basis of spin-spin coupling. So depending on the adjacent hydrogens or protons which are present, so they will split depending on that. So I won't explain it again. Now comes the C13 NMR spectroscopy. So till now we were talking about the H1 NMR spectrum. Now comes C13 NMR spectrum. So in this case, peaks appear as the single lines and there is no splitting, because in the previous case there was coupling which was happening, but in this case we'll only see the single peak. And why it is so? Because there is proton decoupling. And why it is done? So in C13 nuclei can couple with neighboring protons. If we look for the coupling effect in this scenario, so then there will be splitting of the spectrum into multiple such short peaks, and which will make our spectra complicated. So to avoid it, we simply perform proton decoupling. So protein coupled spectrum when decoupling is not possible or decoupling is not done. So in such case, we will observe multiple peaks due to heteronuclear coupling, because there will be multiple adjacent hydrogens which are linked to that particular carbon and its adjacent carbons. So they all will bring spin-spin coupling. So we will see multiple splittings and that will make the spectrum complicated. So in such scenario, we need to perform the proton decoupling, which means now we will remove all such protons and will not see the coupling effect of those protons. So in such scenarios, the carbon will only show a single peak, so which is represented here. In this case, there are multiple such short peaks as you can see here. So the spectrum was not clear enough. Whereas now we will only see only a single peak, and this TMS is the reference to which we measure the change in the chemical shift values. This is the reference sample. So proton decoupled means all nuclear coupling disappears. So single peak is visible and it makes the spectrum easier to analyze. So that is the reason. And this is the distribution. So in the previous slide also, in the previous slides, we have seen that what are the different factors which were influencing the chemical shift values. So in this case also, hybridization, electronegativity, all those things, they influence the how the chemical shift value will vary. So if the hybridization state is sp2 or sp, then it will have the higher chemical shift values and it will be towards the downfield. Whereas if CH3, CH2 alkyl groups are present, then they will be towards the upfield. That means low del values. And another thing which you should keep in mind, that C double bond O, or the more the electronegative groups which are present, or the aromatic groups if they are present, then all these groups they bring about the de-shielding effect and which will move our chemical shift towards the downfield. Yeah, these are the further examples of C13 NMR spectra. So in this case also we will firstly identify that how many type of unique carbon molecules are present, and then we will see that, so in this case, because we will not look into the splitting, so we simply check that how many unique carbons are present. So in this scenario we have 4 unique carbons, so we will see 4 different peaks. For example, in case of acetophenone, so this is a little bit of complex example. So how we find unique carbons, or in the previous case when we were looking for hydrogen, so the first thing is that we need to check that to which groups it is that particular carbon is attached to. So if that carbon is attached to same identical groups or same distance of the other groups, then that carbon will be considered as only single carbon. That is not means considered as the different carbons. So in this case, this particular CH3 carbon, it is attached to C double bond O and it is different from all other carbons which are present. So this will give one peak, so which is given by this green color. Now comes C double bond O. So in one side it is attached to CH3 and on the other side it is attached to a benzene ring. So this carbon is also unique. This will also give one peak. Then comes this carbon which is present in this position. So this is also attached to different groups from all the sides and no other carbon is attached to those functional groups. So this will also give a separate peak. Then comes these ortho carbons which are present on these ortho positions. So both of these carbons, uh, they have the similar groups which are attached on both the sides. So that's why they will only give a single peak. Similarly, these other two, these carbons are also attached to identical groups on both the sides. So they will also give only one single peak. And then this terminal carbon, it will give a unique peak. So in this case, we have one peak coming from this carbon one peak coming from these two positions, from these two meta positions, and then one single peak coming from this ortho position from both these carbons, and then from this para position. So there are 1, 2, 3, 4, 5, and 6. So there will be 6 different peaks which we will observe in this scenario because there are 6 different types of carbons which are present. Okay, now till now we have only talked about the one-dimensional NMR spectra in which we are only talking about the one such element or one such atom which we are considering, either proton or either carbon. But now if there are two different directions if they are present, so that is known as 2D NMR spectra, in which we are taking spectra on the both the directions. So when we are comparing among spectra on both the sides, x-axis and y-axis, with the chemical shifts forming a grid-like spectrum, that is known as 2D NMR spectra. And peaks in the grid correlate to nuclear spin interactions. So it can either be between two H1 or it can also be between H1 and C13. So in this, it tells you about the interaction between both the atoms which we are considering. So there is some kind of spin interactions or coupling which is taking place. So this is just to look for the coupling in which two hydrogens are showing the coupling effect. So diagonal peak corresponds to 1D NMR peaks, while the cross peaks, so the diagonal means this H1 is interfering with H1. So this is just the same. So we will not consider the diagonal peaks. We will consider the cross peaks because cross peaks will tell you that which of the two different hydrogens which are having the coupling influence on each other. So in this scenario, H2 and H1, so they are showing the cross peak, which means H2 and H1, their nuclear spin are interacting with each other. So COSY means correlation spectroscopy, in which both the axes represents the H1 NMR spectra and cross peaks reveals the coupled protons. And this coupling effect is brought about the bond interactions, which means only those hydrogens will show the coupling effect which are separated by only 3 bond distance. So this is only by the bond interaction, and this is not happening in the space. And this technique helps to identify which hydrogen atoms are coupled. So in this case of COSY peaks, we can see that which two groups are lying next to each other, which hydrogens are influencing each other. So this can tell us about the position of those hydrogens in the real molecule because they are giving us spectra. Yeah, this is another example. So in this case also, the D is interacting with D, whereas cross peak is observed in the case of CH3 and OCH2. So this one CH3, which is this, sorry, B and A. So because they are lying next to each other, they are adjacent to each other, so they will show a cross peak, which means they are separated by a bond and they are showing interaction in their spin coupling or interaction in their nuclear spins. So this is about the 2D NMR spectra and the COSY, which is through bond interaction. Then there is another terms such as nosy and rosy. So till now we have only seen the bond-based interactions, but nosy means, so in nosy it is based on the principle of NOE, which is nuclear over-Hauser effect, which is caused by the dipolar coupling between the nuclei. So local field at one nucleus is affected by the presence of other one. The result is the mutual modulation of resonance frequencies. So the intensity of interaction is function of distance between the nuclei. So for simplicity, we will see the example. So in this scenario, this particular red hydrogen is interacting with the yellow hydrogen. So they are lying close to each other in space, but if we look for the bond separation, then they are separated by these 4 different bonds. So which means that there is some kind of coupling which is also happening due to their closeness in the space as well. Whereas if we talk about this green hydrogen and this red hydrogen, then they are lying far away from each other. So in this case, the coupling effect will be very low. It will be nearly zero. So in this way, the intensity of interaction, it is inversely proportional to the distance. So this is the formula. Intensity is equals to A into 1 by R raised to power 6, where R is the internuclear distance and A is the scaling constant. So there is inverse relationship. And nuclear over-Hauser effect spectroscopy, which is known as NOESY, whereas ROSY means rotating frame over-Hauser effect spectroscopy, which is similar to NOESY, but the initial state of magnetization is different among both of them. Yeah, so from this slide, you have to keep one thing in mind, that NOESY and ROSY, You can pronounce it as rosy also. So nosy and rosy, they are the showing the space interactions between the two atoms, such as in this case we are considering between the hydrogens. So this is not separated by bonds, but they are separated by space. And as the distance increases, the intensity will also reduce. Now comes heteronuclear spectra. So in the previous case, we were only looking at the homonuclear spectra, in which we were comparing the interactions among two same hydrogen atoms. But what if we compare among the hydrogen and carbon or hydrogen and nitrogen? So that is known as heteronuclear single quantum correlation coherence, in which the both the atoms which we are considering, the coupling effect which we are considering among the two different elements or two different atoms. So it is widely used biomolecular NMR to study the proteins, because in proteins, the NH, CH, they are interacting with each other. So these are different atoms which are interacting. So it identifies the amide groups in amino acids, serving as a fingerprint to monitor the structural changes or ligand binding. So for example, in this case, we have the amino terminal end of the amino acid. So this is one particular amino acid, and it has the hydrogen which is attached to the amide group. So amide hydrogen represented by HN, and this is the nitrogen. So amide hydrogen and nitrogen, they are also interacting. Their nuclear spins are also interacting with each other. And if there is some ligand binding, then this will change the spin, or this will change the interactions which are happening among both of them. So with this way, if we know the what is the chemical shift before the ligand binding and what is chemical shift after the ligand binding, so there will be change in the chemical shift. So this will tell you that whether the interactions or the ligand which is attached to this, means whether the ligand has influenced the spin. So that can give you the fingerprint that where the group is attached in the amino acid. So this is the purpose that why we go for the heteronuclear NMR spectra. And this is the example. So in this case, W7 is a ligand which binds to calmodulin. So in this case, the black dots represents the calmodulin protein, and the red dots represents the calmodulin protein which are bound to ligand, which is W7. So now there is difference among the positions where the N15 and H1. So this represents the N15 and this represents the H1 peaks, and the points where these means red dots are observed, which means where the interactions or spin coupling is observed among N15 and H. So there is change in the couplings, so change in the regions where the coupling is happening, so which shows that during binding there is change in the chemical shift values. So this is how we can find out the changes, that what are the changes which means comes when there are change in the interactions or when the molecules are different. Second type of heteronuclear 2D NMR spectra is HET-COR, which means heteronuclear correlation. So in this case, it is uses the H1 NMR on one axis and carbon 13 on other axis. So this is also the same. In the previous case, we have looked into the amide hydrogen and the N group. And in this case, we are comparing among hydrogen and carbon. So in this case also, we will observe the peaks at the positions where the carbon and hydrogens are interacting with each other. So this is based on the bonded interactions. So the carbon and all the adjacent hydrogens which are linked to that carbon, so that will that will show the coupling with the carbon. For example, let's take the example of this fourth molecule, fourth carbon. So the fourth carbon, so this shows the hydrogen NMR spectra, and the x-axis shows, y-axis shows the carbon NMR spectra. So in this case, if we consider the fourth hydrogen, so fourth hydrogen is showing the interactions with the fourth carbon. That means the carbon fourth, it is linked to fourth hydrogen. So means the hydrogen present in the fourth position, or it's linked to the fourth carbon. So that means the spin coupling is happening only where the bonded interactions are present. So where the carbon is lying next to the proton and not with this other hydrogen which is lying on the fifth position. So this is what is the difference between the heteronuclear correlation and the previous one. Till now we have discussed 2D NMR spectra. So there is also the 3D NMR spectra. So now previously we were only observing among the two different heteronuclear, or means we were only having two different axes, but now we can have three different axes in 3D. So they can be heteronuclear or they can also be homonuclear. So in this case, it is H1, NH, N15 and C13. So in all the three directions we are seeing that Means if they are lying next to each other, how they are influencing the spin of each other, whether they are bringing about coupling in the spin. So that is known as 3D NMR spectra. So 2D spectra like NOESY and TOXY of larger proteins, they are often crowded with signals. So therefore, those spectra are spreading out in third dimensions so that the signal are distributed in cube instead of plane. And this spread out is achieved by combining HSQC and NOESY or HSQC or TOXY in a single 3D experiment. And then another is the triple resonance experiments. So in this case also, triple resonance experiments are the method of choice for sequential assignment of large proteins which are more than 150 amino acids long. And these experiments are called triple resonance because there are three different nuclei. For example, hydrogen, carbon, and nitrogen. So there are three different nuclei, and resonance means how they are influencing the interacting, how their nuclear spins are interacting with each other. So that is why they are given the name as triple resonance. So how they are correlated with one another. Such experiments are known as triple resonance experiments. And advantage of these experiments is that they have high sensitivity and simplicity, and they contain only a few signals on each frequency. Often there is one signal, but the problem of spectral overlap is therefore markedly reduced. And disadvantage of all the triple resonance experiment is the necessity of doubly labeled protons and the preparation of which is expensive. And then another example of 3D, so depending on that which of the means how the spin is coupled, it can have different names. So one such name is HNCO experiment, in which the magnetization is transferred firstly from this hydrogen to amide to nitrogen, and then from nitrogen to this carbonyl carbon, which is the beta carbon. So in this case, because the magnetization is passed on from H to N and then to CO, so that's why it is named as HNCO, because magnetization is transferred in that order. Similarly, we can also observe an HNCaCO, in which magnetization is transferred from hydrogen to nitrogen, and from this nitrogen to C alpha, and then from C alpha to carbonyl carbon. That's why it's given name as HNCA and CO. So it shows you that how the magnetization is transferred. Similarly, we can also observe HNCA, in which magnetization is transferred from H to N and then from N directly to C. So in this case, because in triple resonance experiments, we are observing through three axes. So in this case, the three axes will be H, N, and C. In this case also, they will be H, N, and C from this C double bond and not C alpha, because it is jumping from this C to C carbonyl carbon. That's why we will not be able to capture the interaction of this particular alpha carbon. So in all these three cases, we will be having the three different axes, which will be H, N, and C. And the only difference which lies in their names is the directionality of the transfer of magnetization. Okay, so now the main thing comes the secondary structure predictions from this NMR parameters. So after you got the NMR spectrum, how will you go to predict the secondary structure? So NMR based structure determinations does not require the full 3D structure analysis as it was in the case of X-ray crystallography. So chemical shifts of amide and the alpha protons provide key structural insights. So there are three bond coupling constants between the intra-residual amide and the alpha protons, which is crucial to determine the secondary structure. And the coupling constants, they're related directly to the backbone dihedral angles. So based on this dihedral angles phi values and the coupling constant, which is JH alpha HN, which is in the units of Hertz. So based on that, we will be able to determine that what kind of secondary structure is present. So this is the key properties. So in case of alpha helix, you will see that there will be sequential NOEs, sequential curves will be present in the 3D spectrum. In N15 edited NOESY are the indicative of alpha helix. So means this is how the coupling constant will tell you the what will be the possible secondary structure, that particular secondary structure which is present in that molecule. Similarly, in case of beta sheets, across strands, NOEs are observed in 3D, and 3D N15 edited NOESY and 3D C13 edited NOESY are indicative of beta sheets. And these are some of the distances. And the another way with which we can predict that whether it is a alpha sheet or it is a alpha helix or beta sheet. So that experiment is done by Wishart et al. He said he has produced a simple method for secondary structure determination by analyzing the difference between the alpha proton chemical shift for each residue and that for the random coil. So in this, we will only compare the difference among the alpha proton chemical shift and the random coil. So this is the difference in the, this is representing the del C alpha. What is the change in the chemical shift of C alpha with the random coil? So if it is negative, that means that region will take a beta sheet. If that particular del C alpha is positive, that means it will adopt a alpha helical structure. So similarly, based on these values, based on the chemical shifts with the random coil versus the C alpha carbon, C beta carbon, we will be able to determine that what will be the secondary structure which is present. What is the secondary structure of the protein which we have considered. Now moving from secondary to tertiary structure. So protein folding is computed using the empirical data because in NMR we get the idea of bond lengths, angles and amino acid sequence. So we have a computer program which refine the structures to match the experimental determined interproton distances because we need to make sure that what are the differences which we are observing from the experimental data, whether it is also matching with what we have theoretically, what are the distances which we are already know that what is the spacing between each atom. So that thing we need to refine. Energy minimization is performed, which ensures that the computed distance it aligns with the expected values. So for that, we perform simulated annealing. In simulated annealing, we search for the global minima by supplying some external energy so that our protein structure, which we have observed, which we have got after experimental analysis, it is in the global minima state. And then NOE distances, they classify the atomic interactions. So if NOEs are very strong, that means it is between 2.3 to 2.5 angstrom, that it means there are strong constraints. That means the structure which we are observing, it is rigid structure. And if NOE distances are weak, which means 3.5 to 5 angstrom, then it is flexible interactions. So goal is to find the stable global minima protein structure. And this is the NOE distance constraints for the structural calculations. So depending on the NOE distances, we can analyze that whether the structure which we are observing, whether it is strong, or whether it has the structural constraints or whether it is a flexible region. So shorter NOE distances means the structure is stable, it's properly structured. Whereas if there is weaker NOEs, the weaker distances, so it means less value of NOEs, then it represents the flexible or the distant atomic interactions. And this is the representation of simulated annealing, that there are some global minima which are present. So to avoid getting trapped in local minima, in simulated annealing, we constantly supply some external energy and we increase the temperature so that we can move to the global minima. So this is how we can identify the tertiary structures by looking at the NMR spectra, by comparing it, by refining the distances, and ultimately the structure which is predicted, it can be means uploaded to the PDB. It can be submitted as we have seen in case of X-ray crystallography. And now if we compare among NMR and X-ray crystallography, so till now the structures which have been predicted by X-ray crystallography, they are very much as compared to NMR. So NMR have not predicted that much structures, but the advantage of NMR is that we can see the movement of that particular protein because it is not rigid in the form of a crystal. It is flexible and it can easily move as it move inside the body. So this is the comparison table. So if you are interested, you can go through this table, and the key points which which are the key differences among NMR and X-ray crystallography are that in case of NMR, in this case the proteins are in the solution state, whereas in X-ray crystallography the proteins are in the crystalline state, which means they are fixed. So due to which we get only the static structures, whereas in NMR we get the dynamic information. So that's why in case of NMR only we can get the structures of intrinsically disordered proteins, whereas in case of X-ray crystallography, this protein had to be ordered. And we can only perform X-ray crystallography if we can crystallize that protein, which is quite time consuming. So this is the main difference between NMR and X-ray crystallography. And now we will quickly go through the MCQs. So first question is, which one of the following EM radiation is of the lowest energy? So option A is X-rays, option B is gamma radiations, Option C is the infrared radiations, and option D is the radio waves. Okay, so without wasting any time, I'll quickly go through these answers also. So among all these radiations, the lowest energy one is the radio waves. If we look at the electromagnetic spectra, then we have seen that the radio waves, they have the longest wavelength and they are low in energy as well as low in frequency. MRI is based on which of the following techniques? Option A is neutron diffraction, option B is NMR spectroscopy, option C is X-ray diffraction, and option D is cryo-EM. So this also we have discussed that MRI is based on the principles of NMR spectroscopy. As in case of NMR, the sample is placed within the magnets, which are superconductors, which are generating a magnetic field, and then we supply the electromagnetic field to bring about change in the spin. And then ultimately the detector is detecting the NMR spectrum, what is the chemical shift which is coming. So similarly, in case of MRI also, the principle is exactly the same. In this case, the human body is kept in the magnetic field, and ultimately, because in our bodies protons are present, so how the spin and chemical shift is happening, that is being captured and MRI image is generated. Third question is, low frequency waves will have option A short wavelength, option B long wavelength, option C high energy, and option D none of these. Yeah, so this is also simple. We have already discussed that if there is low frequency, which means they have long wavelength, because the relationship between the frequency is that frequency tells you that how many oscillations are taking place in 1 second. So one oscillation means one wavelength. So that's why if the frequency is low, that means wavelength is very long. Next question is, which of the following is NMR inactive element? So in this case, the NMR inactive element is C12, because in this case it has even atomic number and even atomic mass. So in this case, that's why it is inactive, and this is the list of all other, that what are the isotopes which are NMR active. So this is the list. Next question is, which of the following is not a factor affecting the chemical shift in NMR? So option A is hybridization, option B is inductive effect, option C is absence of a chiral carbon, and option D is resonance. So you have to tell which of the factors. So this also we have discussed that there are four main factors which influence the chemical shift. And in this case, the absence of a chiral carbon is not directly influencing the change in chemical shift, whereas hybridization, inductive effect, resonance, and hydrogen bonding, these are the four factors which affect the chemical shift in NMR. So C is the right option. Now next question is, which of the following solvents cannot be used in NMR spectroscopy? Option A is water, option B is CCl4, option C is CS2, and option D is CdCl3. So the right answer is H2O. And why H2O can't be used? Because if we use H2O as a solvent, so it already contains hydrogen. So in that way, it will interfere with the NMR signals. Whereas in case of CCl4 and CS2, we do not have any hydrogen. So there are better solutions, better solvents to be used. Whereas if we talk about CdCl3, so in this case, we have deuterium. So deuterium will also not interfere with the proton NMR, in which we are looking for the 1H NMR spectra. So that's why H2O is the right option here. Next question is, why C13 coupling is not observed in NMR spectroscopy? So option A is low abundance of C13, option B is C12 is in NMR inactive, so it does not contribute to NMR signal. Option C is C13 have 400 times weak signal than H1, and option D is all of these. This is also straightforward because we have already discussed that C13 is a very low abundant isotope of carbon. So due to which the signal which we observe, they are very weak, and C12 is inactive, and C12 is 99.99% abundant. That is the reason why C13 coupling is not observed. Question number 8 is, in C13 NMR, the carbonyl carbon will give the signal in the range of 6 to 8 ppm, 20 to 40 ppm, above 150 ppm, or 100 to 130 ppm. So in this case, we have a carbonyl carbon. So the right answer is above 150 ppm. And based on this distribution, we can clearly figure out that the carbonyl carbon is the one which is giving the highest chemical shift, because it is attached to oxygen, which is electronegative, and then double bond is also present. So this brings about the de-shielding effect. That's why it has the greater value of chemical shift in terms of C13 spectra. So next question is, which of the following is a space correlation NMR method? Option A is HSQC, option B is HMBC, option C is ROSI, and option D is inadequate. Okay, so the right answer is option C, which is ROSI. And nosy, if nosy would be there, then that is also right. So if we talk about HSQC, then it is through bond interaction. In HMBC, this identifies the long range correlations based on J coupling and not on space correlations. Whereas in ROSI, it is based on the space interactions. Similarly, because it is based on the nuclear over-Hauser effect, making it space correlated method. And if we talk about inadequate, then it is used for the C13 corrections. Yeah, so that's why ROSI is the correct option. And this is how the rosy peaks appear. So on both the sides, it tell you the where the means space correlations, where the interactions are happening. So these regions will tell you about that. So this is for the C13 and C13. Now the last question is, in which of the following methods direct one bond correlations are suppressed? Option A HSQC, option B HMBC, option C rosy, and option D inadequate. So in this case, we need to tell the methods where the direct one-bond correlations are not observed or they are suppressed. So the right answer is HMBC, because HSQC is directly depending on the direct one-bond correlation. So they are not suppressing it. Whereas if we talk about HMBC, then it is heteronuclear multiple bond correlations. So it is based on the long range interaction. So when there are long range interactions, that means the two molecules are lying away from each other, but there is some kind of bond which is separating them. It is not happening in the space, but it is the long range bonded interactions. So that comes under HMBC, which is suppressing the direct one bond interactions. And if we talk about rosy, which we have discussed, so this is the through space correlations, and inadequate has nothing to do with this. So this is all about all the questions. Yeah, and this is how the HMBC peaks appears. Now I have kept some add-on concepts. So if you are interested, then you can go through these concepts, in which I have discussed that if we combine both the X-ray crystallography and solution NMR, then that can give us better results. Yeah, and there are some key points which I have kept here. So looking at time, don't have much time to discuss them. Then I have also talked about the solid state NMR. So till now we have only discussed about the solution NMR, but there is also another NMR which is solid state NMR which exists. So which is generally used for the cases where we are not keeping the protein in solution, where we are observing it in the means rigid state or in the its natural form. For example, if there are some proteins lying on the membrane, then we are not taking out those proteins and then observing in solution. We are simply observing them at the membrane surface. So this is about the solid state NMR, in which we are observing it in the non-solution environments. Yeah, and another thing is that there are certain proteins which are insoluble. So for that case, we require solid state NMR. And then the slides tell you about why we should perform solid state NMR. So if proteins are insoluble or aggregated, then we perform solid state. If we want to look for the structural details, as I have talked about the membrane proteins and lipid bilayers, and it also overcomes the size limitations because the solution NMR is only limited to the smaller proteins. And in this case, we can use as large protein as possible. And it also resolves the structural heterogeneity. That means when the protein is lying in different conformational states, so all those states can be captured. And then molecular interactions can also be studied in solids by using solid state NMR. And then there are certain papers or reviews which discuss in detail about solid state NMR. And then there was another paper which talks about, so this is a review which talks about that how the solid state NMR can be used in the drug delivery field. Yeah, so this is it for today's session, and so sorry it was quite long. So if you have any doubts, then you can ask. Maybe from the next session, I'll try to keep the sessions short so that we can have more time for discussions instead of rushing through the content. Okay, so if you don't have any doubts, then I'll end the session. And thank you very much. And if after going through the slides, if you still feel some doubts, then you can ask in the discussion forums. So from there also I can answer you. Yes, thank you very much. I'll see you in the next week.